Bismillahirrahmanirrahim. The process of spermatogenesis is discussed in this video. The process of formation of a sperm is termed as spermatogenesis. During embryogenesis, the primordial germ cells they migrate into the testes, and these germ cells then begin to uh, become spermatogonia, which are immature germ cells that are located in 2 to 3 layers along the inner surface of the seminiferous tubule. At the onset of the puberty, the spermatogonia begin to mitotic division to proliferate and differentiate into mature sperm cells. This image is the microscopic cross section of the seminiferous tubule from the testes, illustrating the key structure that are involved in spermatogenesis and testosterone production. So seminiferous tubule, these are the coiled structure that are inside the testes where sperm is formed, or the center of the tubule. It shows developing sperm cells moving toward the lumen for eventual release. The Leydig cells or the interstitial cells, the Leydig cells or interstitial cells, they are found in the spaces between the seminiferous tubule, and these cells they produce the testosterone, which is essential for male sexual development and regulation of spermatogenesis. So spermatogenesis occurs within the seminiferous tubules, and testosterone is produced by Leydig cell located outside the tubule but within the testicular tissue. So this is the structural organization of the testes at cellular level. Spermatogenesis occur in the seminiferous tubule and initiated during active sexual life, that is around 13 years of age, and this continues throughout life and decline in old age. This is stimulated by gonadotropin hormones from the anterior pituitary, and first there is activation and hormonal stimulation. So that is triggered by follicle stimulating hormone and other gonadotropins. The hormone stimulates the seminiferous tubule to begin the sperm production, and then the immature germ cell begin the process. They migrate between the Sertoli cell toward the central lumen to the seminiferous tubule, and these Sertoli cells are large supporting cells. Their cytoplasm envelops around the developing germ cells, and these cells nurture and protect spermatogonia as they develop into mature sperms. The spermatogonia that cross into the Sertoli cell layer become primary spermatocyte. These cells are larger, and uh, they prepare to undergo meiotic division. They undergo meiotic division. Then each primary spermatocyte that contains 46 chromosome, it is uh, divided into two secondary spermatocytes, and each having 23 chromosomes. Then there is a second meiotic division where each secondary spermatocyte is then quickly divided again to form two spermatids, totaling four spermatids from one primary spermatocyte. So spermatids undergo morphological changes, not division, morphological changes to become spermatozoa or sperm, a process called spermiogenesis. The significance of this process is that the 46 chromosome of the primary spermatocyte are split into two groups of 23 during meiosis, and this ensured that each sperm carries half the genetic material, that is 23 chromosome, which combine with the 23 chromosome of the oocyte from the mother to form a complete set in the zygote. The entire process of the spermatogenesis from spermatogonium to mature spermatozoa takes approximately 74 days to occur. This diagram is showing the seminiferous tubule, the cross section of the seminiferous tubule, that is the key structure in male reproductive system that is responsible for spermatogenesis. The spermatogenesis, that is the process of sperm cell development. Spermatogonium are the stem cells that are located at the periphery of the tubule that initiate the spermatogenesis, and the primary spermatocytes are the cells that arise from spermatogonium, and these primary spermatocytes undergo the meiosis 1 and form secondary spermatocytes. Secondary spermatocytes proceed to meiosis 2 and they form spermatids that are the haploid cell formed after meiosis 2, and they mature into spermatozoa, the mature sperm cells that are found near the lumen of the tubule. Then there is this special structure, the Sertoli cell, and the Sertoli cells are supportive cells that nourish developing sperm and help regulate the process. So various cell division occurred during spermatogenesis. Initially, there are primordial germ cells. These primordial germ cells originate before birth and migrate into the testes during embryonic development. And the spermatogenesis starts at the onset of the puberty or around 13 years of the age. So these immature germ cells, they begin mitotic division after puberty and they are converted into spermatogonia, and they proliferate via mitosis on the basal membrane of the seminiferous tubule. Then after about 25 days, the spermatogonia, they are some spermatogonia, they differentiate into primary spermatocytes, and these primary spermatocytes will then enter into meiotic division. The time duration for this is 25 days, and then the first meiotic division occur in primary spermatocytes, and each primary spermatocyte is then divided into two secondary spermatocytes, and these two secondary spermatocytes have got the time duration of about 9 days, and then each secondary spermatocyte is then converted into spermatid containing haploid number of the chromosomes. This takes the secondary spermatocyte conversion into spermatids takes about 19 days, and then morphological changes or differentiation changes, they occur in the spermatids and they mature into spermatogonia or mature sperm. So total time from spermatogenesis to mature sperm is approximately 64 to 74 days. This process of spermatogenesis is highly regulated and stepwise, and it involves mitosis, meiosis 1, that covers the primary spermatocyte conversion into secondary spermatocyte, and meiosis 2, that is the conversion of secondary spermatocyte into spermatids, and finally differentiation, that is the achievement of morphological changes that lead to formation of mature sperm. So each spermatogonium contains 23 pairs of chromosome, one of which is the sex chromosome pair X and Y. The X chromosome is associated with the female and Y is male associated chromosome. So during meiosis, one spermatid receives the X chromosome, becomes a female sperm, and the another is the Y chromosome that becomes a male sperm. So this male sperm can produce a boy and this female sperm can produce a girl. So the sex of the offspring is determined by which type of the sperm fertilizes the ovum. So if the X bearing sperm fertilizes the ovum, that will result in XX, so a female will be born, and the Y bearing sperm, if it fertilizes the ovum that contains X, it will result in XY chromosomal architecture that will result in male. Thank you for watching this video. If you have any query, please write in the comment box.